assemblers. We are very excited to have him speak today, and without further delay, I give you Sergey Nurk.
Thank you very much for such a great introduction. I want to tell you in advance that you will probably be disappointed in this talk, because you probably came to hear about all the new exciting, science and biology and genomics that we can now see in the complete human genome. But this talk will not be about that, because I'm the worst person to discuss that, because I'm not really a great specialist. What I will be mostly talking about is the assembly process itself, like why it was hard, what did we do to make it easier, and how we, pushed to the very end. And then I will give you a brief kind of overview of what's going on, but you really should take a look at at our paper and at the companion papers, which actually second wave of them were released yesterday on bioRxiv, and they are amazing. And my goal for today is kind of hook you up to actually go and read them. And but if you are interested in the genome assembly algorithms and related stuff, then I hope you will enjoy that talk. So first of all, you probably heard that human genome was finished 20 years ago. There were these two papers in Science and Nature. There was a big event in the White House, but unfortunately, it's not true. And even our own institute, National Human Genome Research Institute, kind of hid that a little bit from the general public. And even recently on the website, it said that even in the early 2000s, we got like 99% of the genome finished, but they didn't say that this was 99% of eukaryotic work. So there was also this, very repetitive, hard to assemble regions such as centromeres and rDNA arrays, and actually entire acrocentric short arms. So we did know almost nothing about what the sequence is on the short arm groups of 5 human chromosomes. And then, so overall, roughly 8%, depending on how you compute this, like between 5 and 10, let's say that, of the genome was missing or incorrectly assembled. And why does it matter? Well, first of all, the variation in the regions that we don't know the sequence for is greatly underexplored, because we usually use read mapping to explore variation, right? And if we don't have the reference, it's hard to do that. And then when you have gaps and issues in your reference, it actually can lead to all sorts of different artifacts of the downstream analysis when you do variant calling, for example. And there is an entire now dedicated paper on the bioRxiv telling you, why using this complete assembly is better than using the current state-of-the-art reference. But let's save it for the end. And also, the other part is we don't know what we don't know. I mean, we didn't even know the organization, of many of those arrays. And this is, interesting biology, which can have all sorts of implications. So obviously, like, organizations of centromeres is pretty important for the cell cycle and for all sorts of pathologies. So, and we didn't know very well, how they are organized and how they evolve and so on. And then the new era of sequencing came recently. You're probably aware of that. And it started, probably started in earnest with respect to the, our ability to finish really hard parts of the mammalian genomes, it started with nanopore ultra-long sequencing. And so we now are able to generate very long reads. And I think that 1 megabase limit has been shattered already. So I think the longest read that has ever been seen is on the level of like 2 megabases. And they're great. They are very long. Unfortunately, they are not so accurate as you would maybe want them to be if you worked with Illumina sequencing for a long time. But they are improving steadily, and now there have been announcements about the Q20 chemistry, and the variant and the base calling algorithms improve a lot. And so now you will be able to get reads which are, something like 98% accurate, I think, on average. But then, unfortunately, in some regions, so the errors will be sequence specific, and in some regions, the reads which originate from there will be somewhat lower quality. But anyway, outstanding length, which allows you to span all sorts of repetitive structures if they are, something like 100 KB long, several hundred KB. So what we can do with them, so this is the ideogram of what genome assembly looked like when it actually, was released, human genome assembly. And here the changing colors are not the bands that you're probably used to, but those the change in colors from gray to black shows the ends, the boundaries of the contigs. And you see that assembly was actually very fragmented, and the N50 was less than a megabase. So in 2020, if you get a lot of ultra-long ONT reads, you put them into the state-of-the-art assembler, this is roughly what you get. So much, much better. And so understanding that this progress is coming, my PI, Adam Philippi, along with Karen Mika from University of California, Santa Cruz, they organized telomere-to-telomere consortium to try to actually, finish the entire complete human chromosome. And they were planning to do it more or less one at a time at that point. And they focused on this CHM13 cell line, which is interesting because it's it's not a human being. That's a kind of downside. It's a cell line. The upside is that it's effectively homozygous, so it alleviates the problems of assembling essentially two different copies of every chromosome. It has a female karyotype, so there will be no discussion of chromosome Y in this talk. But since then, we actually have been working a lot on chromosome Y, and I've been working on it a lot in the past couple months. And I should say that I've become even more of a feminist during that time, because frankly, chromosome Y sucks. And if you can live your life without that, I now think that you should, because it's very complicated, full of all sorts of junk, which is super hard to assemble. But we are almost there, if you're interested. And what they did, they started generating all sorts of different data types for this cell line, and they're actually publicly available, which is a cool thing, because now whatever algorithm you're developing or whatever software you want to test, you have all these different sorts of all these data types from this exact cell line, and now we even have a reference for that exact genome. So it should be a nice resource. But obviously we didn't have genome back then, but they had all this sorts of data, and they put it to good use. And they were recently this. So they were able to completely assemble human X chromosome from this cell line, and then human chromosome 8. And this was done by Glenys Loughlin in Evan Eichler's group mostly, but we actually also helped because it kind of overlapped with our work, and we already had some draft reconstructions of chromosome 8, and it kind of was a very cool partnership between us and Glenys, because she pointed us to some errors that we had, and we pointed her to some errors that she had, and overall it was great. So both of those reconstructions were mainly based on laying out the ONT reads. So they were mostly ONT based, and it made it hard to reconstruct some of the, for example, centromere. And Karen spent a long time of like semi-manual process to piece it together. Actually, it's very interesting. So they suggested a way of doing that using marker k-mers. And actually, they observed that within the centromere, there are variations. Oh, actually, they didn't use marker k-mers. They used structural variations. So they saw that in this array, sometimes you have a couple more copies over short repeats. Sometimes within the unit, sometimes you have a couple fewer copies, and you can catalog these variants and then look for them in the ONT reads and use them as kind of markers, for joining the ONT reads together. But it was pretty painful. Since then, it has been this or similar process has been automated by Pavel Pevzner's group in UCSD. Check out the Centerfly publication, for example. But with with the newer techniques, it became somewhat less pressing, because it now seems that we can do a easier and better job from hi-fi reads. I will tell you how. And what are the hi-fi reads? So our first, cool data type was ultra-long ONT. Our second cool data type, which I will focus mostly for the rest of the talk, is PacBio hi-fi reads. And PacBio hi-fi reads are actually, if you know about CCS reads that PacBio had, those are the same thing, but rebranded, because so they now can achieve the length of the dozens of kilobases, so up to 20 kb. And before that, if you ever worked with CCS reads, they were like 800 base pairs. And although they are, much shorter than ultra-long ONT reads, which can span like hundreds of kilobases, they are very accurate. And so mean read quality is something like 99%. But the most importantly, I think, is that with this level of noise, we can make the reads even much more accurate. And I will tell you more about that, and that's what actually helped. But even before we came along, people have shown that they are useful for improving variant detection and assembly, but they were showing the results which were kind of on par with what has been previously achievable, at least for the assembly part. And then, when I joined the lab for Adam Philipp's group, my first project was to try to adapt the Canute software to kind of better deal with hi-fi reads. And we were able to do exactly that, and we published Canute last year. And the key observation kind of was that if you can work with those reads, they're great. But if you do some work and make them even more accurate, then when it really starts to pay off with respect to assembly of very similar regions in human genome, or haplotype separation, or satellite array analysis. So we did that by self-correcting the reads, and I will show you example later, and handling of some systematic errors. So if you are used to PacBio slogan that the errors are purely random and that's why they're better than everybody else, it's no longer true for hi-fi reads, at least. So there are systematic biases that you better take care of, otherwise they will introduce, gaps and problems. So yeah, human assembly. With respect to human assembly, the highlights of our paper was that after doing that, HiCanoe can achieve accurate allele separation and long, perfectly phased blocks. We have shown improved assembly of segmental duplications and even draft reconstructions of centering. And this is where we started thinking that maybe we can, now kind of refocus on PacBio hi-fi reads as a faster way to get to the complete human genome, because that's what our lab was very, very interested in. And so we developed this custom telomere-to-telomere assembly pipeline. If you are interested in more details, check out the methods for our bioarchive preprint. Also, there are some scripts. There is a repo, but nothing is documented yet. So I would suggest you not to look there. But if you're interested, ask me. But I will also tell you why you probably shouldn't be using those methods and should wait a little while before we come up with something really decent. This was based on actually experimental code that I developed for us to, as a sandbox to play with different kind of assembly approaches and to be able to debug HiCanoe, but it, branched into its own thing. And so the pipeline overview is the following. First, we do hi-fi reads correction and overlap search. Then we construct the assembly graph, which is very simple, which in this case is actually Myers string graph. And then we do somewhat manual reconstruction of genomic paths. So we need to identify genomic paths through this graph. And then we can, after we identified the paths for the chromosomes, for parts of the chromosomes, we can compute consensus from hi-fi reads and do assembly. So first things first, read correction and overlap search. So similarly to HiCanoe, as I mentioned, we do homopolymer compression because most of the errors in hi-fi are, this variable lengths of the homopolymer runs. So instead of 3 Cs, it might have 4 Cs or 2 Cs. So we just compress them to, a single letter. Then we do self-correction based on the 99% overlap. So we find the overlaps between the reads, and then we look at the pileup of the reads. And if none of the reads essentially supports that there should be A here, and vast majority of reads says that there should be T here, we correct this base pair to a T. And then we recompute overlaps, and then we mask differences in microsatellite arrays. And this is where these systematic errors of PacBio hi-fi are really concentrated. And we didn't know how to deal well with them because you will see reads, that support different copy number of those microsatellites. And so we just decided to kind of ignore those differences when we score and when we search for overlaps. And it worked great actually. And after that, so after we kind of rescored and we ignored those differences, we then retained only perfect overlap. And at this point, you immediately see that actually our pileup that I've been showing you actually consists of 2 groups of reads, which probably originate from 2 copies of very similar repeats. And that's actually the power of hi-fi reads, that you can distinguish the kind of the origin of the read, even though those repeats that the reads originate from are very, very similar. And so here you see that one of them has G in this position and another has A. And after we see that, we essentially will not consider the overlaps, right, between the first two reads and the third and fourth read in the graph, which will kind of, automatically untangle those repeats. And effectively, we won't see them as repeats within the assembly process at all. And so then we construct assembly graph. For that we run patched version of mini-SM to build my string graph. And we use minimal overlap size of 1 KB. So what's a string graph? Essentially, your vertices are reads, and then you put the edge between the two reads if they overlap, right? And then you just compact the non-branching paths. So if you have only one way, one read essentially that extends you forward and one read that extends you backward, you can join them in a single node. And so we don't use any further mini-SM processing. And then instead we use custom pruning procedures. We remove tibs, bubbles, usual stuff, more or less, low coverage nodes, etc. Then we use progressive threshold to remove weak edges. So which correspond to overlaps which are below certain threshold. And we progress up to 8 KB. Why I'm telling you this, it's because in the end, the graph that I will be showing you, you need to imagine that every edge that you see corresponds to essentially a perfect repeat of 8 kilobases. And all the smaller repeats, they are just resolved within the graph automatically. So another note is that it's all in home polymer compressed space, right? So when I say 8 KB, it's actually not 8 KB of the original read, but 8 KB of the home polymer compressed, which will correspond to something more like 11 KB uncompressed usually. And then we remove non-genomic edges in certain simple subgraphs. That's so some heuristics on removing some of the edges that we definitely don't need. And then in the end, we did minor manual adjustments. Already while we were doing reconstructions, we realized that some of the nodes were removed. We did not need, which we didn't want to remove, or some links edges were missing, and so we brought them back. And another thing that I wanted to highlight is this part about coverage, because it's something that actually hasn't been part of the standard string graph based assembler toolkit. And the reason for that is that it wasn't that obvious how to estimate coverage for short nodes, which correspond to kind of just a handful of reads. And we found a way to do that, and we used our experimental approach, and it really helped us. And I will show you later how it helps to remove some of the erroneous nodes, but you can immediately understand that it can be useful, for example, for understanding the multiplicity of the nodes. And for example, in this case, you immediately see that you need to pass this loop, traverse this loop only once. And so this is the graph that we got after a couple rounds of fixes and feedback from our colleagues. And actually, it's pretty interesting. So there are multiple observations that we can make about that. And first of all, so in gray, we mark here the centromere associated arrays of repeats. So like alpha satellite arrays are there and human satellite arrays are there. and maybe some other types of satellites. So most of those, hard regions to assemble, most of those tangles, they are in the centromeric regions, which can be, multi-megabases long. And another interesting thing is that, most chromosomes are their own connected components, right? Or there are some breaks sometimes, which are marked in triangles here, and they actually correspond to the GA-reach microsatellite arrays, which are, somehow hard to sequence with for PacBio HiFi, and so you just don't have read coverage there, or reads are very inaccurate, and so we needed to patch them later. But overall, if you color by aligning those nodes to the sequences of those nodes to the reference and color by the chromosomes that they align to, you don't see many places where they are different chromosomes are connected. Kind of the only exception are the junctions of rDNA arrays, and this is actually this gives you an idea that there were some recent recombination events there, which, if you know something about rDNAs, apparently shouldn't be surprising. I didn't know much before, but now I know a little more because my boss is very, very interested in rDNA arrays. So, right, so essentially it means, and it's somewhat surprising, but it means that there are no, perfect repeats longer than, say, 8 kilobase, like 10 kilobases on different chromosomes, except for rDNA flanking junctions. Also, there are no distant links even within the same chromosome. And so they are all mostly look, linearly, except for some well-localized tangles. And the exception to that is that huge tandem duplication on chromosome 9, which actually, as I now learned, is probably a rare kind of variant in human population. So most of you probably don't have that, but yeah, we had some troubles dealing with it. But there are some very complex tangles. There are rDNA arrays on the short arms of acrocentric chromosomes. There are HSAT arrays on chromosomes 1, 4, 6, 9, 16, and they don't look pleasant. And so we had this. And then we decided to start a T2T summer finishing workshop. And it was, 2020. We started planning it, early, I think late winter. We started inviting people. They started to say yes or no. We had some capacity. We were planning to gather them all on NIH campus for like a week. And then obviously coronavirus pandemic started and everything went online. And so instead of a 10 days long workshop, we had like a month-long workshop working nonstop with, dozens of people all across the world. Nobody could say no at this point already because they didn't need to travel. Online meetings were still new, so people probably didn't want to evade them. So much. And we had multiple working groups. I obviously was in kind of the production assembly working group, but also there were other working groups and people worked on the parts that they were, mostly interested in. And most of them now resulted in their own kind of, papers analyzing this genome. And so during the workshop, we've been doing this manual genomic paths reconstruction, which I will tell you a little bit about now. So first of all, in this, in the perfect case, you want to traverse, so you want to find paths that traverse all nodes. And so you don't want to skip nodes. You can skip edges in these graphs. That's totally fine. And interestingly, the candidate path is often unique, even with this simple constraint. Well, you also add constraints that some of the nodes you want to traverse only once based on their length, for example, but we also have coverage estimates that further help. So for example, here you immediately understand that you need to traverse this loop once. And here you understand that actually none of those nodes are repetitive, because otherwise they would have coverage which is higher than coverage of the sink and source and sink of this tangle. And so you need to traverse them once, and there is only one way to do that, which is cool. Then coverage also allows you to ignore some obvious artifacts, which were not ignored by the kind of manually set threshold, because in different regions the coverage might be somewhat different. Also, the appearance depends on the actual sequences that are there. Anyway, there are some artifacts that we can ignore. Also, we saw quite a few heterozygous variants, even though it's effectively haploid cell line, it's still the real cell line, and it accumulates some differences between the copies of the chromosomes. And also there might be some variation, some SNVs, for example, within the population of the cells, right? So we saw events like that, and we chose, usually the most common variant, or if they were, the same, and roughly the same coverage, we chose the longest one. And then for the complicated tangles, actually, an iterative approach worked kind of nicely. So if you look at this example, you can imagine that you immediately, understand that you have the only path. But another way to look at that is that you look at the edges, one at a time. And sometimes you can understand that some of those edges actually cannot be used in any genomic work. For example, if you use this edge, then to get into this node, you will need to traverse this long node the second time, and you are not allowed to do that. So you can remove it, right? The same for this edge. And then you keep doing that. And this argument allows to resolve some pretty complex tangles, even without any additional information. So, for example, this tangle on HSAT, this HSAT1, I think, on chromosome 1, you can resolve it to this stage, at which point it's obvious that you need to get here, traverse this loop, get out, and you're done. This was quite unexpected, so I thought that it will be much harder, and somehow when you learn your sequence assembly, everybody tells you that you use string graphs, it corresponds to Hamiltonian path finding, you use like Debrun graphs, it corresponds to Eulerian cycles, but then people say that it doesn't matter in practice at all, because the repeat structures are usually give you so much ambiguity that you just extract your unitics and you're done. But apparently, on this at this level of resolution for reads of this length, it's not like that. So actually, Hamiltonian pathfinding can help you with multiples. But then some HSAT reconstructions are somewhat the best guesses. So some of the regions are less reliable than others, but in general they're very good. So there might be there have been occasional potential problems flagged within the HSAT arrays, but those were on the order of like there was a handful of them, like on the order of, I don't remember, like 5 maybe for the entire for the entire genome. Right, but there were some cases which couldn't be resolved just from hi-fi reads, obviously, because they are short after all. And there we integrated ultra-long ONT data that we had. And interestingly, a lot of the cases looked pretty simple, like this hairpin, for example, but you can do not understand the orientation of this last bit without some additional information, because hi-fi reads just don't give you any hint on what you should do here, or here the same thing. And for these simply looking things, actually aligning reads onto the graph with Graph Aligner, developed by Mikko Rautiainen, who is right now in our group and is working on kind of novel methods for genome assembly. It worked really nice, and it gave us enough evidence to understand the orientation. There were, more ambiguous. And I should say that we didn't have kind of, automatic resolution. So we actually looked at the alignment logs, and results, and actually, went to Bandage, which is an awesome tool for visualizing those graphs that we've been using extensively, and chose the correct path. Obviously, right now we have it all automated, but that was almost a year ago. So, and we didn't want to wait. But in more ambiguous or difficult cases, we actually, generated multiple alternatives going through the localized region. We then aligned ONT reads with minimap, winomap. We then, for every read, considered the identities of the best versus second best alignments. And we picked informative reads where the best alignment was, considerably better than the second best. So we thought that, okay, this read probably hit some unique area and is actually informative for understanding, which path is correct. And then we inspected evidence from those informative reads also, manually at this time. And this part is not automated still, but we hope that we won't need it soon. And then it allows you to pick the one of the traverses. So, and then 2 words about acrocentric chromosomes. So I told you already about this recent recombination that gives you the only place in the genome where different chromosomes kind of intertwine in the graph. But the cool observation was that you see those like very complicated blobs, right? And those actually correspond to RDNA clusters themselves. And cool thing that you see immediately is that, well, on this picture, there are 4 of them, but actually there is a pit somewhere disconnected. But there were 5 of them, which immediately kind of suggested that, the RDNA arrays are not so well homogenized between the chromosomes. So, and we indeed, like with further analysis, we found, the events specific to, individual chromosomes. And that was pretty cool because that was kind of unexpected. We were expecting that, they are always homogenizing kind of within the genome. And so they will all be very similar, all like 400 copies of them. Well, in haploid, like 200 in haploid. But it wasn't the case, which was a very unexpected and interesting finding. But it took like another half a year to actually get to a decent draft reconstructions of those arrays. And even those are still, they don't account for all variants and somewhat model-based, but we can discuss it later. But here I just wanted to show you that with ONT data and ONT data, we were able to kind of, untangle those this intertwining between the chromosomes. And then we were able to, with some high C signal, which was not easy to get, we were able to verify that we associated correctly the telomere with the century, essentially of those, of those chromosomes. Right. Okay. And then came time for consensus and assembly post-processing. So we directly translate the parts that we got into backbone, hi-fi reads, backbone chain of hi-fi reads, because that's what's easy to get from string graph. Then we place the nested reads. Then we uncompress the reads, because I remind you that everything happened in the compressed home polymer compressed space. So this is actually the place where we uncompress and actually get the real underlying sequence. And you have to be careful with coordinates, but we already had it more or less figured out while working on HiCanu. And then you can compute consensus. And then we had to tweak it heavily to improve accuracy in highly repetitive regions, in particular centromeres. And we had amazing feedback from Alla Mikheyenko and Andrei Vizikadze from Pavel Pevzner's group, who had developed before that, to our joy, they had a tool for validating centromeric arrays based on ONT data, we happily it was very, very helpful for this. And then we had to patch 25 gaps, which were there due to this, GA dropout. So in the GA reach microsatellite regions, the coverage of hi-fi just drops, sometimes to 0. And here you see coverage, for different libraries, and it's always 0. And ONT doesn't have any troubles with that. So we used this observation and patched the assembly by, the previous ONT-based assembly that we had, because though these regions were no trouble for ONT-based assembly, and then we obviously polished and repolished it. So it was polished before, we polished it again. And the maximal gap size was almost 7 KB. Then one of the telomeres had to be patched, but since then we found it in updated version of hi-fi graph. And then we had a polishing team, and they were also validation team, led by Aran Gree from our group, and they did an awesome job finding variants, cataloging them, looking at the evidence, and then introducing changes to the assembly. And so they polished essentially from Q68 or 69 to more than Q70. So it's 7 nines worth of quality after the, so it's 1 error per 10 megabases, give or take. And they had to introduce just a few structural few bigger corrections. I think 4 of them. And then half a year and rDNA arrays later, which had to be, so Miko had to develop all new method to get to the reconstruction, which I won't be describing here. But now we are happy to announce, the complete sequence of this genome. We don't have Y chromosome, but we have other 23 chromosomes. Nothing is unlocalized, nothing is unplaced. There are no gaps, no known structural variants. We're at Q70. And we added roughly 200 megabases of new sequence to the current reference. A lot of it is in acrocentric chromosomes, so the entire short arms of those, as I said before, were missing. So we added 60 megabases base pairs of sequence there, but also quite a few regions like in centromeric regions were missing on chromosome 16 and 1 and 9. So it's all cool. And then everything is released to the public. So if you're interested, this is a nice kind of point to access those resources. It has the graph that we've been working on, the paths that we chose, version 1.1 gapless and polished assembly. It has links to the raw data and other useful links. For example, links to the preprints that are getting released related to the analysis of this assembly, and also genome browser. Yeah, this is all available now. We also have draft annotation, but don't judge us too much. Although we collaborated with the experts, but there is only so much you can do without 10 years of curation, kind of of this. And yeah, a lot of new interesting biology is being discovered. So this link has links to the preprints, and also there is a lot of commotion on Twitter. I would recommend you to check out Adam's Twitter, and he has a pinned tweet Twitter thread where he collects all the related papers, but then obviously also the authors of the papers. They usually provide you an extensive summary of the new and exciting findings. So from our side, we looked at we're mostly looking at so from the interpretation point of view, we mostly were looking at the alpha-centric chromosomes, and my colleagues have revealed all these, exciting structures of satellite arrays. Essentially, the entire, so here is the entire short arm of the 3 chromosomes, and it's all satellites and RDNA array. So RDNA array here, then you can see the color code below, like this is HSAT1, beta satellite, HSAT3, alpha centromeric alpha satellites, and so on. We were very proud when we discovered one new type of satellite which hasn't been kind of known before. So that was interesting. But then cool thing about that is that if you look at this scheme below, you will see, and then at each of the chromosomes, you will see that the overall structure is actually similar. The way that those satellite arrays follow each other is kind of defined. And it's also interesting, if you look here, you will see that it's kind of symmetric. So you have this acro, then SD, then HSAT3, then beta satellite, then HSAT3, HSAT1, and then again beta satellite is HSAT3, and so it's like a palindrome. But the lengths of the each of those satellite arrays are very different across different chromosomes and within the same chromosome. So you see HSAT3 is huge on chromosome 15 here, but then you can hardly see it on chromosome 14 and 13. So yeah, interesting stuff about the evolution of those. Now a little more about the preprints. I will put the links on the slides when I share them in some PDF format, probably. So we had a group, amazing group led by Justin Zook and Mike Schatz. who were analyzing how using the new reference would affect variant calling, both with long reads and short reads. And across the board, they see that using this complete reference is much better than using the original, the GRCH38. And for the details, please look at their paper. Also, we had there was an amazing group led by Winston Tim, who dedicated their work to looking at epigenetic patterns in the complete human genome. And now that we have sequence, then Evan Eichler's group was looking at the segmental duplications, and a lot of them we've never seen before. Essentially, we added, as much, this assembly adds as much segmental duplicated sequence as there was in the reference before that. So kind of double. Also, in this ideogram, you see that, interestingly, a lot is shared between the acrocentric chromosomes and other acrocentric chromosomes, but also between acrocentric chromosomes and other chromosomes, which is quite interesting. And on the left, you see, segmental duplications marked within the same chromosome. So it's within chromosome, that's across chromosomes, but it's super interesting. Also, Mitchell Volger has looked at, how the copy number of segmental duplications in this genome corresponds to the Simon's copy number, segmental duplication copy numbers across Simon's diversity panel. And this information is available on the UCSC genome browser. So that's also a super valuable resource. But also one of the interesting results of this analysis is that T2T, so CHM13 is remarkably unremarkable. So it's kind of, very kind of close to the. So if you try to come to create the reference that would, kind of best correspond to as many of different genomes that we have analyzed. Actually, CHM13 will be pretty close to that kind of, best possible one. And GRCH38 will be way, way far. So, some of the segmental duplications will have very different copy number in GRCH38 than in effect, actually, more or less any other genome that you look at. And it's likely an artifact of GRC, of, errors and GRCH38 being just incomplete. Also, Karen Miga's group has been focused on analysis of everything related to the centromeres, and they just yesterday released an amazing preprint about evolution, structure, and comparisons to other humans, chromosome X centromeres. And it's just amazing. Check it out if you are interested in this topic. This is kind of, probably will set a new standard for the field. But I also liked Nick Altemaz's tweet a lot, where he said that it's, similar to how we got a first good photo of Pluto. And before that, we saw, just the blurry images, but now we see in high resolution. And he compared this genome to that. So before that, we had kind of model-based reconstructions of some of the centromeric regions, and now we have, the entire sequence, and they can look at where, at its structure and evolution, and where the actual protein are attached to. Yeah, so they're pretty excited. Then there was a group dedicated to analysis of other repeat elements across the new genome, and so check out their preprint as well. And yeah, so now about kind of plans going forward a little bit. So stay tuned for the work of Human Pangenome Reference Consortium. If you think that just one genome is not enough, you are right. And they will they are committed to have very high quality reconstructions of 200 plus deployed genomes. They won't be T2T, at least yet, but they are of very high quality. And also, so we hope that this work will be able to spark kind of a movement for getting T2T reconstructions for model organisms, because you might be surprised, but we don't have complete reconstruction of fly chromosomes, so mouse chromosomes as well. And so we hope to kind of spark this initiative and also help getting there. Some summary thoughts. So finally, at least one human genome is complete. And it's the most bases ever added to the reference. We added 5 new chromosome arms. There are some new genes, which are vast majority of them are paralogs of the known genes, but still. And new structures in the genome are uncovered. CHM13 is a across the board better reference than what we have right now. Unfortunately, it might take community quite some time to do the transition because of the inertia, obviously. Also, PacBio is a super powerful data type, and most of the vertebrate genome sequencing projects that you will hear in the coming years will most likely be based on PacBio using the additional data types to resolve some residual long repeats and fill the gaps and do the phasing. So what's next for kind of T2T consortium? We actually need much better assembly methods. Well, much but better assembly methods. So this pipeline that we've used here has some inherent downsides that we can discuss a little during Q&A. And also it requires required some, well, not too much, but still some manual input that we don't want, and we don't want to pay this price for every new genome, and it will be much it might be harder for deploys. And also the model itself, kind of the string graph model, does not fit well for the deployed genome assembly. To be honest, not saying that somehow PacBio data that has been generated recently has been somewhat worse than the ones that they generated before. Luckily, I think it's a temporary thing, and that the redistribution will become tight again at some point, but maybe not for all organisms. And string graphs really suck when the read length distribution is kind of wide. So anyway, that's more or less it. So many thanks to Sergey Koren, who has been my mentor, and who was my partner in crime in reconstructing those untangling those tangles. Brian Wallens, who is an engineer in our group and who has helped us along the way to adopt the method's extensive GNU code base to our will. And Adam Philippi, who has been an inspiration and has guided us along the way, and all the others. So this is the key players kind of our consortium, but there were so many more people there too. And yeah, if anybody is still there, I can take any questions.
Yes, of course we are here. Thank you very much for a really lovely presentation, Sergey. I
You're welcome.
actually have a couple of questions. I have a couple of questions on my own, but before I will move to those. I will read a question from German Demidov.
Oh, hi, German.
German is actually asking about Y chromosome. So he says that there are several assemblies of the Y chromosome, even in T2T. Were you thinking to collaborate with authors of those papers? And is it possible to simply include their assembly, or do you think it must be chromosomes from the same cell line? He also adds that he saw some criticism that because of the missing of the chromosome Y, your assembly is actually not full.
Well, it's not exactly a valid criticism because we claimed assembly of a particular human genome, and this human genome has XX carrier as half of the Earth's population, more or less. So, but it's indeed a very good point that for using this thing as a reference, you need to have some chromosome Y draft. So for the analysis that I know about, people have been using just adding the chromosome Y from GRCH38 to the consideration, and it worked well, as good as it could, kind of. Chromosome Y reconstruction is very incomplete. Like tons of megabases of sequence is just missing. From there, I think I had, let me see, I think on the very first ideogram there was kind of it was showing how many ends are there, and you will see. But while I'm clicking, I wanted to say that unfortunately this cell line doesn't have chromosome Y, right? So we would be happy to get it from here. So what we are actually doing, we are working on reconstruction of, yeah, here it is. We are working on reconstructing chromosome Y from a different cell line, which is also widely available. It's AG002 genome from the Ashkenazi trio. And also there are available, like a lot of different data types are available. I think it's part of the Genome in a Bottle consortium, but I'm not sure. But we already have a draft reconstruction of chromosome X for this cell line, and actually it was used by some of the companion analysis papers, and we now have essentially we improved it and we'll release it soon, so we have complete chromosome X there, and that's the cell line which we will have chromosome Y for also in the upcoming months. Whether I don't know, I also saw those criticisms, but luckily this one is quite easy to address. Like if this is the main criticism that people have, then we hope to have draft reconstruction of chromosome Y from HG002 by the time the paper is out from the review, essentially. So stay tuned. We are almost there. It will be harder for non-sex chromosomes of HG002. So we're working for pipelines for T2T kind of deployed assembly, but we are not there yet. But we, at the same time, we started working on a panel of sex chromosomes from different kind of human genomes. So we hope that we might have a panel of chromosome. So we will definitely have a panel of chromosome X T2T reconstructions for dozens of humans. We hope to have chromosome Y for some or most of them, but we will see how it goes. Chromosome Y is a major pain, but again, we're almost done. So it's a pain that I've already suffered.
Thank you very much for that answer. And and German is also thanking, so I guess he's satisfied with with that. And I just want to tell you that there are many comments on YouTube that are just thanking you for your great presentation. But I'll move to a question from Anna Maria now. She says that great work and the talk. And homopolymer compression is a very interesting approach. However, I guess it reduces the specificity of the initial read alignments. Did you try to estimate this somehow?
No. We should have, but we kind of were, so occupied after initial HiCAN work. We were so occupied with kind of the production of this genome, and now with other practical genomic projects that we kind of did not return back to this. So it's very true. Indeed, we might kind of miss actual true variation, not just in homopolymers, but also I mentioned that we ignore differences in microsatellites, right? And we might lose some kind of resolution there for sure. So far, it seems that in practice, it's not that big of a deal, right? So the fact that we were able to resolve those, maybe in some of those cases where we had to use ONT data, we wouldn't have if we accounted for this homopolymer differences. That might be true. A little unlikely, but so a matter of fact, I would say, is that even if it would help in some cases, in some it won't. So you still need kind of integration of other data types. How much it would help, I don't know, because we had the way to integrate other data types, and we just dealt with all the problems across the board. But it's a very good question, and it would be nice to take a look. Somehow, I mean, my understanding was that, luckily the primary mode of this small scale evolution are SNV, are point mutations, right? Substitutions. That I might be wrong. But so our hope when we were, implementing that was that if you have some differences within the region, you will have some point mutation as well, right? Some substitution, and that would be enough. In cases where you only have homopolymer differences on the in the range of 8 kilobases, like 10 kilobase pairs, there we indeed will not see it, and we could have used it if we were smarter, but we are stupid.
I don't think that's the case. Seriously, I don't think that's it. I think you're smart given your presentation, but yeah. So I think thank you very much for that answer. And there's another question also from German. He's asking whether we should start aligning our clinical data to T2T reference, or should we still use HD38 and wait a little bit longer before transition?
German, I'm afraid you will have to, just because probably all the databases, right, that you have use this reference. I would refer you to a very interesting discussion on Twitter in Mike Schatz's Twitter account, where they are discussing, what would be needed to do this transition and what would be the perks and benefits of that. I mean, definitely read their preprint. I hope you will be convinced. So they lay out a quite convincing argument why it should be done. But whether it can be done, how easily it can be done, it's a great question. Great help would be if like maybe like we still don't know what will be the actions of Genome Reference Consortium, to be honest. So in what way or form will they adopt some of the regions or entire chromosomes or entire assembly? We don't know. In any case, we kind of hope that soon we will probably transition to something like pangenomic references, where we will have a panel of dozens of very, very high quality human haplotypes. And in this, at this like worst case scenario, at this time, this will just be one of them. Before that, really read the paper, read their Twitter, ask there, discuss. I'm not a good person to discuss that because I don't know the clinical genomics world well. So I don't know.
The clinical genomics were just started switching from from HG19 to HG38. So I guess we are like 10, 15 years.
No, I mean, but maybe it's a good point. But maybe it's a good timing since everybody wants to understands that they need to switch. Maybe they can switch to a different one. Maybe instead of switching to HG19, you can just switch to HM13 straight. But really, I have no idea. But what I know is that I think if you see some, weird results, maybe it would make sense to redo the analysis based on CHM13 because some of them might go away, especially since AG38 had actually introduced some problems. They introduced some duplicated genes that are actually not in this chromosome. I think for chromosome 22, they kind of placed one of the backs incorrectly and introduced, extra copies of some genes that are just not there, which might give you troubles. Also, AG38, for example, has quite few copies of some of the paralogous genes, and they don't have kind of they have pseudogene versions, which leads to incorrect, like way more reads piling up where they shouldn't, and showing you all sorts of variants which are actually not there just because the reference is missing the other bits. But again, read the preprint and participate in the discussion. I don't know what will happen.
So I actually have a question of my own. Sorry for the drilling because today is just the worst day ever. But yeah, someone is renovating their apartment upstairs, and there's nothing I can do.
No worries. We hear you well.
Okay, apologies. So my question would be, first, it might be a very naive question because I'm not really an expert in assemblies. But when you showed the photo, like the new photo for chromosomes, it made me think, what about the spherical, like what about the 3D orientation of the chromosomes? Do you think that can influence somehow the ease or how hard it is to assemble? Because we do have a lot of work done in the space of the chromosomes being close to one another, some regions being hard to detangle because they are so close that sometimes we sequence them as one read. Do you think that kind of influenced your assembly in some way?
The answer
Or is it
no.
just naive?
Apart from, as I mentioned, apart from this case with the acrocentric chromosomes, where we see that there were some recent recombination, and this is probably for good biological reasons, and also has to do with them being kind of co-localized most of the time. Apart from this case, we did not experience any influence of the regions from different chromosomes being close. Maybe we would. I mean, with the previous methods, and it was actually quite surprising that the chromosomes were divided kind of so well. Right, but it seems because we knew that there are like segmental duplications on different, like copies of segmental duplications on different chromosomes, which was thought to be highly identical, but not identical enough to confuse the new methods and new reads, right? So maybe if we were looking at the lower resolution, maybe it would affect what we would see, but at this resolution, it did not. Also, right, yeah. So also, for example, these HSATs, right? So there are like HSAT, same type of HSAT, human satellite 3. You have them on different chromosomes, and they were thought to be, very, very similar with each other, but apparently they are not similar enough to confuse us. And even like straight from the initial assembly graph construction, you see that they are diverged enough between each other. So the only similar copies, you see them only within the same chromosome. And copies between different of those higher order repeats, they are different enough between different chromosomes that they didn't confuse us. So yeah, apart from acros, I guess the answer is no.
That's very cool. That makes sense. Yeah. Thank you very much for that. There's another question for Anna Maria, from Anna Maria. So she asks, in your opinion, what's the bottleneck in applying Hi-Fi approach to other organisms? Do you think it will be more readily applied than Hi-C assemblies, which are now most common way to improve genomes?
So the answer, I don't know too well. I think the answer is there is no bottleneck. Really, because even high-scale initiatives like Darwin Tree of Life, which you really care about, having a robust sample preparation methods, and as far as I understand, that's partially the reason why, for example, they don't use ONT data. They actually completely switched for all the new data generation for Hi-Fi to Hi-Fi. Human Pangenome Reference Consortium immediately switched to Hi-Fi after seeing initial kind of results from HiCanu and Hi-Fi SM. So Vertebrate Genomes Project, as far as I understand, most of the new data will be generated on Hi-Fi platform for the time being, maybe until ONT becomes even more accurate, or we learn how to deal with the lower accuracy of ONT. I should say that it's it's not just technology; it's combination of technology and ways to process the data. And it might very well be that as soon as ONT data kind of pushes through some invisible boundary of the accuracy, suddenly our methods that we have and that we understand and we know how to develop well, they will start working for ONT very well. Right, so it might be that there will be next switch, but in the time being, for the next, like right now, big consortiums actually generate Hi-Fi data. It doesn't mean that Hi-C goes away, I wanted to say. So the actual protocol right now seems to be to use Hi-Fi as the primary data set, right? So for primary assembly, and then use Hi-C and then whatever else you have for assembly improvement. But it's cool that you mentioned Hi-C because that's kind of the most popular combination so far, I would say, and kind of very perspective going in the short and midterm. But at the same time, for T2T work, we would prefer Hi-Fi plus ultra-long ONT combination. So far, we will focus our efforts on and we'll encourage others to put their efforts into organisms where we can generate ultra-long ONT, but it's a bottleneck about ultra-long ONT and not Hi-Fi. As far as I understand, you can generate Hi-Fi for almost everything these days.
Yeah, that sounds really cool. And how is it about the price? Because from what I understand, ONT is quite price attractive. Like it's quite low cost. Is that the same for Hi-Fi?
No. So, but kind of good news is that hi-fi is these days, hi-fi is not much more expensive than original CLR. It's not cheaper though.
Okay.
So yeah, it's about what you want. Like, do you want more genomes? For these big initiatives, it's kind of about quality quantity trade-off, right? Do you want more with lower and unreliable quality, or do you want less for with good quality? It's not an easy question. Depends on your goals, I guess, right? But at this day, and like in summer 2021, we can generate a much better assembly from PacBio Hi-Fi than from ONT. And one of the reasons for that is actually not just continuity; it's also about consensus. The thing that I haven't mentioned is that when, for example, my colleagues with Karen Miga were working on chromosome X assembly, right, and they got to the ONT backbone and they got ONT consensus, they had to spend like a non-trivial amount of time to develop a methods that would be able to polish, those highly repetitive arrays based on the kind of not very high quality consensus that they got from ONT. And so it was a task of its own. You don't have this problem with hi-fi. With hi-fi, like straight away you have very, very high quality consensus. And that's another reason. So if you want it's not just about continuity, although you can separate repetitive regions much better with hi-fi than you can with and haplotypes, right? So for example, if you care about haplotype assembly, hi-fi is also your kind of only bet, I would say. I don't know any other technology that could give you a better comparable result. And with hi-fi plus Hi-C or strand-seq, you can get chromosome scale haplotype resolved assemblies. Check out the papers from Ken Lee's group or from Evan Eichler's group. It's pretty remarkable what can be done.
Yeah, that definitely sounds cool. Anna Maria says, yes, they are complementary. I think that's about Hi-C and Hi-Fi, of course. And for genomes where we still have only scaffolds, not chromosomes, Hi-C is very useful. But cool that the long reads are becoming so common. Thank you. So that's from Anna Maria. And I guess I have another question coming back to the clinical genomics. So if I understand correctly, your assembly, the T2T assembly right now, that's done based on one cell line. So what we're going to have is we're going to have a collection of SNPs or a collection of bases that will be specific to maybe not one individual as those are cells, but one kind of set of mutations, like I don't know, not mutations, one set of letters. And then you mentioned also pan-human project. How are you gonna basically merge all those different data, like all those different information into one assembly? Are you gonna include? Is it gonna be able? Are you able to include all those variety in SNPs throughout human genomes into one assembly, or is it going to be?
That's a really cool question. I think that's a work in progress kind of for the community, right? How they want to represent. So obviously for right now you have like alternative alleles within the reference, right? So there are quite a few alts, and people are working on kind of better ways to integrate those and better methods kind of to use those. So all this commotion about genome graphs or smart ways to index and multiple rounds of like alignment to kind of different genomes that you have. I have no idea, to be honest. I mean, that's a tough problem, but as far as I understand, the state of the art, like what we have currently is you have a reference, right, and the alts, and the reference is bad in many regions and is absent in many others, and this is kind of a better reference. That's to be honest, that's not why that's not why I think we were doing this. So probably that's why you can get money for doing something like that. But obviously, our motivation was very different. But it seems that kind of right with the current technology stack, right? You'd be better off switching because you can only right now you can only use one reference, and it better be a good one. In the future stack where you can use multiple references, genome graphs, and so on, I don't know how it will work out. I think that maybe your question was, whether we want to introduce common alleles into this reference, right, instead of minor alleles, and it has something like that. Maybe in the short perspective, it's useful, again, because of the limitations of the current stack that we have. In the midterm perspective, I hope it's not. I hope that people will figure out better ways to kind of provide the access, and analysis against kind of all known variants across multiple genomes that we know about. But again, I'm the worst person to talk about that because I have no idea.
Well, I disagree that you're the worst person, but thank you very much, nonetheless, for your answer.
Sure thing.
I wanted to say just last one remark. I promise. I wanted to say that your photo, like the figure that you made with the chromosomes, with how they look like in the graph space, I think that that's really cool because we finally get an upgrade from the karyogram that we were always showing and when we were talking about chromosomes. So it's really really cool. And
I
kind of
know.
changes.
To be honest, I think the best. Do you mean the graph representation?
I think so. Yes.
Like, sorry. Sorry.
Don't worry.
Do you mean
Yes,
this one
that's one.
or the?
Yes.
But I don't agree with you. This one is useless.
Oh.
That's kind of work in progress because the chromosomes. Yeah, you see, the problem with this figure is that it's very method specific, technology specific, right? So it doesn't reveal to you the repeats that you might care about. I think the much better figure is, for example, the figure from Mitchell-Vogler's paper that I showed about segmental duplication, right? Because there, like you set a threshold on the length of the identity of something that you consider to be segmental duplication that of interest to you. Things that are repeated here, for example, I can tell you why it's a bad picture. Some chromosomes have almost no branchings, even in the centromeric region. So they have a multi-megabase long centromeres, but it's kind of diverged enough within itself that it looks trivially in the graph, although it's not. And whether do you really want to discriminate the centromeres, on the based on the fact that some of them are kind of more diverse within themselves than others? I'm just not sure. So no, I think that's what I wanted. So graph representations for assemblies are useful only, I think, for people who are doing the assembly. But for everybody else, I think that if we succeed, after all, every chromosome is a linear thing. So we should strive to assemble it to the state of being, one linear sequence. And everything else is just beautiful. It's indeed beautiful, and it gives you some insights. But I would discourage anybody to try to get a lot of insights from this, like analyze the sequence. It's now possible. You have it.
Okay, I stand corrected then. I am all for equality. I'm not gonna be against equality for centromeres. They should have also be looked at. So yes, I stand corrected. Thank you very much. I think that's all the questions that we had on YouTube. If anyone wants to ask one, this is your chance right now. I guess that's all. Those are all the questions. But thank you very much. This talk was amazing, and I enjoyed it very much. Despite the fact that I chose the wrong picture to get hyped about, but I will correct myself. I will stand correct. Thank you very much. It was a joy.
Thanks for having me. It was fun.
Yeah, thank you very much. And all of you who are listening, so this was a seminar in our NGS seminar series. We are meeting again in a month. We're meeting on the 18th of August. We're going to be talking about proteomics, and we're going to release the info about the speaker very shortly. And in September, instead of having an NGS seminar, we are going to have NGS Sprint, which is a hackathon event for which you can still register. All the information is on our website and on our Facebook and Twitter social media. So please check it out. So again, NGS Sprint is a hackathon in which we're gonna divide into 4 or 5 hackathons. You can read the descriptions also on our website that we will post links to in the description of this video and on Discord. And please look out for announcements about new seminars. Thank you very much for joining, and see you next time. Bye bye.